I'll start to introduce our next speaker. People, there are still seats available in the front row. Please do come strictly in while I'm doing the introduction. So our last session's first speaker is our very own Dr. Mark Baer. He's the Picower Professor of Neuroscience at the Picower Institute for Learning and Memory and the Department of Brain and Cognitive Science at MIT. He's a man who does not need a whole lot introduction. Welcome.
Thank you, Li Wei. It's so nice to be back at MIT. Anyway. Did you need a map? So, well, I'd have to agree with what Danny said. This has been a mind-altering experience, and really enjoyed the talks today and the breadth of the topic. And I also have to agree with Matt that I stand on the shoulders of giants. And I must say that I was forced out of my comfort zone a little bit in trying to come up with a talk that I thought would be of interest to such a broad audience as this, considering what we spend our time doing in the laboratory. So here it goes. So we all know that there are many ways to mess up brain development, and these include early childhood adversity, disease, and heredity. And so we can conceptualize this in the following way, where we have some measure of brain maturation. Of course, there'll be many different measures of brain maturation depending on what's the process that we're interested in. But in general, there's a trajectory of normal brain development. And in the face of disease or genetics, this development can be derailed, in which case there's an accumulated deficit. And so we've heard a lot of really inspiring talks today about how one might prevent some of the damage that occurs, particularly with early life stress. But the issue I want to address in my brief presentation today is what are the prospects for treatment once a diagnosis has been made or a problem has been identified. So this is the big question we're going to address. And you can imagine really three different scenarios ranging from quite pessimistic to wildly optimistic. So the pessimistic scenario is that by the time a diagnosis has been made and a treatment has been begun, it's really too late. We've missed a critical period that any intervention we can do would be at best palliative and in no way disease modifying. The most wildly optimistic scenario is that the treatment actually corrects the core pathophysiology of the disease, or maybe even replaces genes that are missing that cause the disease. And there's actually a correction of this trajectory of altered brain development so that actually with time and training it can recover normal function. And finally, there's of course an intermediate stage, which I call the hopeful scenario, which is that it's never too late to intervene, and there will always be some benefit, even if that means a reduced rate of decline in function. So today I'm going to tell you about two short vignettes about two projects that we have going in the lab that are relevant to this theme. And what I'm going to convince you is that we can exploit knowledge of neurobiology to repair genetic disorders of brain development and to overcome deleterious effects of impoverished early experience. So one of these is a disease called Fragile X, which is a genetic disease of brain development, and a second is a human clinical condition that's caused by poor quality vision during infancy and early childhood. So let me begin with Fragile X. So Fragile X is a disease that is caused by the transcriptional silencing of a single gene in the genome. So it's a single gene disorder. It's relatively rare. It affects about 1 in 4,000 males and half as many females. And it's a consequence of, as I said, a silencing of this gene called FMR1 and the failure to express a protein called FMRP. And as a consequence of this one failure of genetics is a syndrome that is multifaceted and very disruptive to both to the affected individual and to the family. So originally this was called Fragile X mental retardation years ago because a prominent feature of just about everyone that carries this mutation is intellectual disability. But in addition to intellectual disability and cognitive impairment, there are a number of different comorbidities that you could categorize in a number of different ways. I think it's useful to consider some we might call hyperreactivity or hyperexcitability phenotypes that can include an increased incidence of early childhood epilepsy. Everyone with Fragile X has autistic features, aspects of social phobia, gaze avoidance, eye contact avoidance, repetitive and stereotype behaviors. And children with Fragile X can be identified based on a number of dysmorphic features, such as a protruding jaw or ears that stick out. Fragile X is often referred to as the leading known heritable cause of autism because just about everyone with Fragile X will fall on the autism spectrum, and a third will get the full diagnosis of autism. So the disease itself was first described back in the 40s by two astute clinicians, Martin and Bell, who identified a cohort of their patients that had features in common, most notably intellectual disability, but also other physical features and other behavioral features. And Martin and Bell suspected a genetic etiology. And of course, it took many decades to identify what that etiology was. It required the discovery of the structure of DNA and the sequencing of the human genome. But eventually, in 1991, a group led by Steve Warren at Emory University identified the gene that's affected. And shortly thereafter, a knockout mouse was made. That is, a mouse that carries the same gene mutation, or a gene mutation that causes the failure of expression of this FMRP protein. So this Fragile X knockout mouse has been around now for quite a few years, and it really attracted the interest of two groups of investigators, those who are interested in this disease, Fragile X syndrome, and those who are interested in the protein, FMRP. Turns out FMRP is very interesting. It's highly expressed in the brain and at synapses. And in fact, both the protein and the messenger RNA exist at synapses. And so there has been a lot of interest in the potential role of this protein in synaptic plasticity, which is what we're interested in. And so I fall in that latter category. Frankly, when we began, I hardly knew what Fragile X syndrome was, but I was quite interested in the protein. Well, in the course of studying how synaptic function is altered in the absence of this protein, we made a small discovery. And the small discovery was that a phenomenon we were studying, a form of synaptic plasticity that's triggered by activating a particular type of glutamate receptor, the metabotropic glutamate receptor 5, or mGluR5. We found that this form of plasticity was exaggerated in the Fragile X knockout mouse. And on its own, it was a modest finding, and we shopped it around, eventually published it in the Proceedings of the National Academy. But we were excited by it because we suddenly realized that perhaps many aspects of Fragile X syndrome could be accounted for by excessive activation of this particular neurotransmitter receptor. So this idea became known as the mGluR theory of Fragile X, and we and many other groups around the world have tested this idea. And by 2007, there was a consensus that mGluR5 was a potential therapeutic target in Fragile X. And shortly thereafter, drug trials were initiated and are ongoing right now to test the efficacy of mGluR5 inhibitors and other drugs that probably act on the same mechanism in human Fragile X. So, as I said, we began with an interest in synaptic plasticity, and we were particularly interested in this metabotropic glutamate receptor, mGluR5, because it was becoming apparent that it was part of a molecular machine that ensures that the supply of synaptic proteins keeps up with demand. And in this case, demand is registered by the release of glutamate by active neurons. So the more active your brain is, the more glutamate is released, the more the mGluR5 receptor is activated, and that this activation of this receptor triggers the translation of messenger RNA that already exists at synapses, translated into proteins that are important to sustain synaptic function. And like any machine, there are important control mechanisms. And one of these is provided by this FMRP protein. So if you imagine mGluR5 is like the accelerator on a car, the FMRP protein is like the brakes. So it turns out it's an mRNA binding protein, and it gums up the works and slows the rate of translation in response to mGluR5. So in the absence of FMRP, what happens is that the brakes are missing, so even tapping on the accelerator has exaggerated consequences. And you can imagine how that would disrupt function, or navigating down the Southeast Expressway. And so as I said, that alone was of some interest, but it was really when we started putting all the pieces together, realized that many of that very large constellation of symptoms that goes along with Fragile X many of those could be accounted for by excessive activation of this receptor. And so we wondered if exaggerated consequences of mGluR5 activation provided a thread that would connect these really diverse symptoms in Fragile X. And as I said, this became known as the mGluR theory of Fragile X, and it's based on a simple assumption that many of the neurological and psychiatric consequences of Fragile X are due to exaggerated activation of mGluR receptors, and with one very important consequence. And the important consequence is that if we could dial back signaling through the mGluR5 receptor, we might be able to correct many aspects of Fragile X syndrome. So we set out to test this idea, and perhaps the most definitive way to do this was to cross Fragile X mice with another line of mice that had half the normal level of the mGluR5 receptor. So what we were going to try to do is to see if we could improve the course of Fragile X in the mice by reducing the mGluR5 receptor expression by 50%, and maybe bring the accelerator, take our foot off the accelerator, if you will. And so Gould Dolan did this when she was an MD PhD student in the lab, crossing Fragile X knockout mice with the mGluR5 heterozygous mice to create mice of 4 different genotypes. And what Gould described was in about eight different phenotypes, ranging from anatomical disturbances in the microstructure of the cortex to electrophysiology to biochemistry to behavior, including some simple learning and memory tasks, that merely by reducing mGluR5 receptor expression by half was enough to correct all of these varied phenotypes. So this is good validation of mGluR5 as a target, but of course that doesn't mean it could be translated into an effective therapy, because that manipulation was a germline manipulation. So those animals grew up with half the normal levels of mGluR5 receptors. So we anticipated Fragile X syndrome in those animals, and we were able to really prevent it. So it really left unanswered the question in a human setting, once a diagnosis is made, which can be at 2 years of age, is it still possible to correct this core pathophysiological mechanism to see an improvement? And so to examine this question, what we required was good drugs, particularly a good mGluR5 inhibitor with a long half-life that we could use for chronic dosing studies. And fortunately, our colleagues at Roche not only came up with such a drug, but they were willing to share it with us. So we had a collaboration with a group at Roche led by Lothar Lindemann. And they developed a molecule called CTEP, which is a highly selective mGluR5 inhibitor, but has an unusual property that it's got a very long half-life, an 18-hour half-life. So what they were able to show was that with every other day dosing, you could have a more or less constant receptor occupancy, as it's modeled here in this graph. So we could block about 80% of the mGluR5 receptors with every other day dosing, which is something that graduate students are willing to do. And so the design of the experiment was that the animals were reared normally up to P30 in a mouse, at which time all these symptoms, all these phenotypes that we study have emerged, and then treatment was begun. And as the title boldly declares, we were able to reverse many of these phenotypes. Essentially, all the phenotypes we were able to reverse with the genetic rescue strategy, and then some. So, of course, I highlight my own work, but this has really been a community effort. So the idea that mGluR5 inhibition might help Fragile X was first presented back in 2002. And the community, the Fragile X community, really rose to the challenge to test this idea. And I can tell you it was met with considerable skepticism in the early days, for good reasons. But in no other field of science that I've ever been involved in has there been such a strong consensus. So that the last time I did a thorough review, which was, as you can see, 2 years ago, but already there were 31 different papers published from different groups looking at 40 different mutant phenotypes, and most importantly, in 3 different species. So not just the mouse model of Fragile X, but also a zebrafish model of Fragile X and a fruit fly model of Fragile X. And in all of these studies, they've reported that inhibiting either the mGluR5 receptor or the signaling pathway downstream of the mGluR5 receptor was sufficient to ameliorate mutant phenotypes. So I stress this because, now there's a lot of scrutiny about the failure to replicate many, particularly animal studies, and between labs and so on and so forth. But this is really reassuring. So this was many independent investigators, many who didn't have a stake in the mGluR theory like I did. As I said, different species, many different assays, and a lot of approaches. Not one molecule, not one genetic approach, but many, dozens of approaches. So this is a particularly well-validated target. So some of the lessons that we have learned just doing this work is that one advantage we had in Fragile X over some of these more complex disorders is that it has a defined genetic etiology. So we have an animal model that's valid. It's wonderful to see that fundamental synaptic biology revealed signaling defects that suggested a therapy. So that means, the century of work that we've been doing on basic neurobiology can have a payoff in terms of therapy, in addition to just knowledge. There can be evolutionary conservation of core pathophysiology. It was an extremely important point in terms of, increasing your confidence that you might be able to translate these insights into humans. Disease-modifying treatments are feasible, and treatment can be successful after symptom onset. And I just want to say that I've been talking about Fragile X because this is what we work on, but there are other single gene disorders that are listed here for which others, mainly, have also found that it is possible to do disease modification with small molecule therapies, and even when those therapies are begun after adolescence. So there's a lot of optimism in the field that we might be able to fix these diseases that used to be thought to be intractable. So our work is done, and now we can sit back and let... Well, okay, so this is where we are. So we have an extremely well-validated target, and now all we need to do is complete the circle. Well, it turns out that's a lot harder than you might imagine. And I probably didn't appreciate this going into it very well. But what is required is a lot of good guesses, okay? So, you have to guess what's the right patient selection, what's the right molecule, what's the right dose, what's the right treatment duration, what are the right endpoint measurements, and so on. So with every one of those decisions, no matter how smart you are, or how many experts you convene, it's not a sure thing. And it did remind me a little bit of the Warren Buffett challenge where, he was quite confident that his $1 billion was secure because nobody was going to be able to guess 32 basketball games and get them all right. And of course, he was correct. So, we can do the back of the envelope, literally, calculation, as I've shown here. I should say this envelope was addressed to the Internal Revenue Service. So I guess I missed that payment. But in any case, so this is just off the top of my head, some of the things that had to be guessed right. And I gave myself a pretty high probability that I would guess right. So 0.8 probability for each of these things. But of course, by the time you multiply all those probabilities together, your chance of succeeding in any one clinical trial is low. And this is why it is very expensive to get medications approved. And obviously, you need to be either extremely lucky or prepared to fail before you can succeed. And so I just want to say that, I've been involved in clinical trials. I founded a company to pursue clinical trials. And here's some of the data. And this is a phase 3 trial. If this trial had succeeded, there was a chance the FDA would approve this medication for Fragile X. These are some of the measurements that we guessed would be moved by treatment. We had 3 different doses, and the treatment duration was 8 weeks. And here are the p-values, and all extremely encouraging. I think you could say this is great. If you got this in your mice, you'd be really excited about it, and you'd want to follow up on it. In fact, we achieved statistical significance in one measure called the Aberrant Behavior Checklist Irritability Subscale. But this is a failed trial. And it's a failed trial because this was not declared to the FDA to be the primary endpoint. That was declared to the FDA to be the primary endpoint. and that is not p less than 0.05. So that's a failed trial. So when you read in the media about failed trials, it doesn't mean the target's no good or the molecule's no good or you don't have the right approach, but it means that you didn't get lucky on the guesses that you made. So now I want to shift gears to talk a little bit about amblyopia in the time I have remaining, and so looking at effect of overcoming sensory deprivation. So amblyopia is a severe loss of vision, usually confined to one eye, that's caused by a number of developmental mishaps, including the misalignment of the eyes, or differences in refraction in the two eyes, or a loss of form vision in one eye. That is, one cause would be a cataract in one eye, or another is a droopy eyelid in one eye. And as a consequence of this deprivation of normal vision in infancy and early childhood, you lose visual acuity, and it can be very severe. And over 50 years ago, Hubel and Wiesel introduced an animal model in which we can study this. And so they were, of course, pioneers in looking at the organization of the ascending visual pathway. And in one important series of experiments, they asked what happens if we deprive one eye of normal pattern vision, either with an overcorrecting contact lens or a patch or by merely gluing the eyelids together. And what they discovered was that there's a stereotype choreography of changes in the cortex so that cortical neurons stop responding to stimulation of the deprived eye. And usually, over time, there's a compensatory increase in the strength of the responses to the non-deprived eye. So one way you can demonstrate this is with electrophysiology. So we can put a microelectrode in primary visual cortex. And so what these histograms show are action potentials that are collected in response to showing a visual stimulus to the right eye or to the left eye. And in a normally reared, these experiments were done in kittens originally. In a normally reared kitten, you can see that here's a neuron that responds well to stimulation of both eyes with a slight preference for the right eye. But after a period of binocular deprivation where that eyelid was closed, they opened up the eye, and look what happened. The neuron stopped responding to stimulation of the right eye. And this is just a histogram showing a whole large population of neurons. So it's a very robust phenomenon. It's called ocular dominance plasticity. So what are we going to do about it? So this is a cartoon that Pavan Sinha gave me, which I think is so fantastic. I'm going to read it to you because if any of you are blind as I am in the back row, you'll be able to see this. But so this is I can't remember which character that is. Is that Lucy? No, that's not Lucy. Patty. It's Peppermint Patty. Okay. So Peppermint Patty said, "I suppose you're wondering why I'm wearing this eye patch, eh, Linus?" Linus replies, "You probably have amblyopia ex anopsia. The vision in your right eye is so dim, so the doctor has patched the left one, thus forcing the right eye to work." Actually, treatment of amblyopia is one of the most rewarding in medicine. Without medication or surgery or hospitalization, a child can be given eyesight in an eye which otherwise might have no sight. And then you can see her response. So Linus is a very smart guy. But the ophthalmologists were pretty smart, too, because they realized that it's possible to restore vision in an eye that has been weakened by deprivation by closing the good eye and forcing the weak eye to work. Okay? And so this can be demonstrated electrophysiologically as well. So here's a neuron again. This is now after a period of binocular deprivation. So this neuron only responds to the open eye here. Doesn't respond to this eye. Now what's happened is there's been a reverse occlusion. So the seeing eye is closed, the open eye is closed, the weak eye is opened, and I just want you to think about what happens at that moment. Okay, at that moment, you have a brain that is receiving input through an eye that has been disconnected, and it's receiving no input through an eye that is strong. So essentially, from the point of view of the brain, there's activity would fall precipitously, but nonetheless, over time, there is a recovery of activation by this previously deprived, newly opened eye. And unfortunately, it usually comes at a cost that the previously strong eye loses strength. So a lot of this patching requires a little finesse to get it right, so you don't mess up the other eye as well. But the question is what can we learn about how to make weak synapses strong and promote recovery of function by studying this? And that same phenomenon occurs in mouse visual cortex, and virtually everyone in the field works in mice these days. And so in a mouse, it just so happens that the visual cortex comes dominated by the eye on the opposite side, the contralateral eye. So these are the evoked responses from the contralateral eye and the ipsilateral eye. But what happens if I patch the contralateral eye? Well, what happens is the contralateral eye responses go down, but the thing that's of particular interest here is that the non-deprived eye responses pop up. And it usually takes about 7 days, okay? So there's a delay, but these responses pop up. And one interesting feature in the mouse is that the same thing occurs in the adult mouse as well, okay? So what's going on? Well, here's a couple of features of this open eye potentiation. So this is what happens normally. So I've plotted here the duration of deprivation of the other eye, and this is the response to the non-deprived eye. And you can see that nothing happens for about 3 or 4 days, and then it slowly comes up. So by 7 days, it's about double in strength. Now, this is not merely a homeostatic adaptation because it doesn't occur if you put the animals in the dark. So it's not the absence of visual experience. It requires visual experience to see this change. And it's completely blocked by an NMDA receptor antagonist, okay? Aha. So that's a drug that we know blocks synaptic plasticity. So we know a lot about NMDA receptor-dependent plasticity. And what we know is that NMDA receptor activation triggers both synaptic strengthening and synaptic weakening. And what you get out of an NMDA receptor depends on how strong it's activated. So if it's strongly activated, you get synaptic potentiation, long-term potentiation. And if it's weakly activated, you get long-term depression. And there's a balance point here, or what we might call a modification threshold, where LTD, depression, is converted to potentiation. And we further know that this crossover point is itself modifiable. So this can adjust depending on a number of factors. Okay, so that's what we know about NMDA receptor-dependent plasticity. So perhaps what happens when you do this reverse patching experiment is that you slide the modification threshold to the left to now make possible synaptic potentiation that previously couldn't occur. So we can test that idea. It's quite easy, actually. You can take mice or rats or cats or put them in the dark for a couple of days and ask, does that shift the properties of NMDA receptor-dependent synaptic plasticity? And the answer is yes, it does. And we described this many years ago. So you put the animals in the dark for a few days, and this LTP, LTD curve, if you will, shifts to the left, and it promotes LTP. So that makes this weak, otherwise weak synapse, gives it a toehold to start to undergo synaptic potentiation. Now, there are probably a dozen different mechanisms that contribute to this effect, but one of the ones that we're confident on, we know, is a change actually in the molecular subunit composition of the NMDA receptors themselves. So when you put animals in the dark, the subunit composition of the receptors changes, so there's a reduction in what's called the NR2A2B ratio, subunit ratio, and when the animals are restored to the light, that ratio goes back up. And we know that changing that ratio will promote or inhibit long-term potentiation. So is this related to this recovery of function? Well, it seems to be, because we can now do these simple monocular deprivation experiments in mice where we reduce the expression of NR2A, the NR2A subunit, by either half or completely. And that's what's summarized here is a study that we did some years ago showing now just with 3 days of deprivation, which normally would produce a very modest, non-significant increase in the response to the non-deprived eye. In the NR2A heterozygotes, we see that achieves statistical significance, and it's really gangbusters in the NR2A complete knockout. So manipulating the NR2A2B ratio is one way that you can promote recovery of function or make weak synapses stronger. And in fact, there are companies actually now looking at NR2A specific receptor antagonists to try to do just that, to promote synaptic potentiation. Okay, so this is what we think is going on, is that initially we close the strong eye and we set up a situation in the cortex where it goes quiet because it's seeing through an eye that's too weak to drive a response, and the eye that's strong enough to drive a response isn't seeing. So the cortex is quiet. As a consequence, there's an adjustment, a property we call metaplasticity, a change including a change in the NR2A2B ratio. And eventually, over time, this is sufficient for experience through this weak eye to start to drive those synapses up, and you start to restore a response. So what about treating amblyopia, and maybe by extension other disorders that are caused by early environmental deprivation? Well, it's not, at first blush, it's not quite so optimistic. And in fact, we've known this since the 70s, since the late 60s, again, beginning with the work of Hubel and Wiesel, who showed that the consequences of deprivation diminish with increasing age. And then subsequently, Blakemore and Van Slyters and many others had looked at the effect of reverse patching. And this just happens to be data from Blakemore and Van Slyters done in kittens, but it makes the point that their ability to reverse the effects of deprivation falls precipitously in the cat, so that by 12 to 14 weeks of age, it's really immutable. Okay, there's no recovery is possible. And this is a feature not just of kittens, but also monkeys and in humans. And this is why when you do reverse patching experiments in humans, you've got to do it before the age of 7. Otherwise, it's too late. So here's an example of, in a cat, just showing this meager recovery that occurs. And this is an interesting experiment done by Don Mitchell at Dalhousie, where just with a brief period of monocular deprivation in a young kitten, and then what he did was he opened up the eye and he looked at the visual acuity of the deprived eye and the non-deprived eye. And this is measured behaviorally, not just electrophysiologically. And so what you see is that here's the visual acuity through the deprived eye. You can see it's 0, so the animal is blind. And it does drift up, but it doesn't drift up. It sort of parallels the acquisition of acuity through the non-deprived eye. Okay, so there's no, it's sort of what I call the hopeful scenario. By restoring vision, it does improve with age, but it'll never catch up. All right? So this is consistent with human clinical experience as well. But, you know, I showed you already, if you were paying attention, that in the adult mouse, we're still able to get this really dramatic plasticity. And at first, this took us by surprise. Well, I guess it still takes us by surprise. But the mouse visual cortex is really extraordinarily modifiable, even in adults. And so it begs the question, are mice so different? I just happen to like this picture, so I had to figure out a way to get it in here. But, and the question is, really, they could be different, evolution. But it could be that we just haven't really exploited the mechanisms that are available in the appropriate way. And so what Don did was he did an experiment to exploit this property of metaplasticity. Okay, and so what he did was he took this animal, this is one kitten, up at the age of 60 days, okay, no further recovery is possible, and they put them in the dark for 10 days, and then brought them out of the dark, okay, put them in the dark, quieted the cortex, changed the NMDA receptor subunit composition, etc. Bring them into the light, and now look what happens to the deprived eye response. Over the course of just a couple of days, it soars and it achieves absolutely normal monocular acuity. So complete reversal of the effect of amblyopia. So this is a really exciting finding. So I'm pretty sure my time is up. Well, I hope it is, because that's all I have to say. But just some of the lessons that we've learned from our studies in visual cortexes. Well, first, there is a lot of plasticity in adult cerebral cortex, far more than is historically appreciated. So this is great news for all of us, all of us, that we just have to figure out how to gain access to these mechanisms, but they are present, they are there, and they can be exploited if we're clever about it. The knowledge of synaptic plasticity and metaplasticity suggest approaches to promote recovery of function. And as I've shown you, we can rejuvenate weak synapses by exploiting this property of metaplasticity. But I didn't have time to talk about work that's going on in other laboratories, other approaches, but all of which are based on knowledge of how synapses modify and trying to use that knowledge to promote recovery. And with that, I just want to say thank you to, well, of course, all the people in my lab that did the work. And I want to thank the funding agencies that have supported us. Particularly, I want to point out the Picower Institute Innovation Fund and the Picower Neurological Disease Research Fund. These are creative funding mechanisms that we enjoy here in the Picower Institute, thanks to Barbara. And they really help us to push the envelope in ways that wouldn't be possible with traditional sources of funding. So, thanks.